Can
Could I
everybody
suggest,
see this?
please? Yes. Now I think so. Outline presentation is seen. Thank you.
Okay.
Okay, now it's here. Okay.
Okay. Maybe I can
And,
start now.
yeah.
Okay. We'll be discussing a drug discovery from Nigerian biodiversity, challenges and prospects. By outline of presentation, I will go from discussing the introduction. I'll talk about biodiversity and drug discovery. I'll talk about the current status of drug discovery from biodiversity in my laboratory, look at the challenges, prospects, and then we'll end for today. As a way of introduction, Nigeria, we are talking about biodiversity, drug discovery from Nigeria. Talk about Nigeria. Nigeria has 36 states. It's a country in Africa, of course. That you can see this reporting on the screen. Where I'm speaking from in Nigeria, and it's very close to the administrative capital of Nigeria, which is Abuja. We're estimated to have a population of about 200 million people, and we share boundary with some other African countries. Nigeria has over 300 ethnic groups, over 500 spoken languages in Nigeria. Of course, the official language in Nigeria is English language, but there are three major ethnic groups in Nigeria, and they are the Yoruba, Hausa, and Igbo. Nigerian beauty is not only about the people, but also about nature itself. Nigeria has a very, very wide and varied biodiversity, starting from the mangrove swamp in the south, which borders the Atlantic Ocean, and it goes on like that until you get to the arid savanna area in the far north. Talking about biodiversity, of course, we know biodiversity is gotten from two words, biological and diversity. All the variety of life that can be found on Earth, that is plants, animals, fungi, and microorganisms, as well as the communities that they form and the habitats in which they live, is what we call biodiversity. The Nigerian biodiversity refers to the variety of plants, animals, fungi, and microorganisms that are either indigenous to Nigeria or that are found in Nigeria. So when we'll be talking about Nigerian biodiversity, we are talking about those that are found in Nigeria or those that are actually indigenous to Nigeria. But for the purpose of drug discovery, biodiversity is considered as those biological species that are studied specifically for their value as producers of small molecules. These small molecules are also called products or secondary metabolites. Now, biodiversity is a very, very rich source of potentially appreciated molecules or natural products. The terrestrial plants have been the most and longest studied sources, having afforded a large number of natural products that have been used for treatment of various diseases dating back to historic times. The discovery of penicillin from Penicillium in the 1930s introduced microbial sources for discovery of natural products in drug discovery. And then this was followed by slow but steady progress in discoveries of natural products from marine organisms. Talking about drug discovery, drug itself are natural products or synthetic chemicals that can alter the way in which the body works or can be used to prevent or treat diseases. Drug discovery is the process by which new candidate medications are discovered. Historically, drugs were discovered through identifying the active ingredients from traditional remedies or by serendipity. Substances were screened for biological activities without the knowledge of the biological targets. Only after an active substance has been identified was an effort made to identify the target. In modern drug discovery, however, the identification of screening hits, medicinal chemistry, and optimization of hits to increase the affinity, selectivity, metabolic stability, and oral availability is the medical team. As one of our board members, Holy Saul, stated in one of his books, the concept of antigen and antibody and venom and antivenom in nature has shown us that nature or biodiversity can be the solution to all problems, because we see from that concept that for every problem we find in nature, the answer is also in nature. And talking about drug discovery, from nature now, from biodiversity, we start, the normal process is to start from extraction. You extract using an extractive medium, and extraction can lead to fractionation of the fractionate, you purify. From purification, you can then do structure elucidation. When you have elucidated the structure, then you have a pure compound, a pure characterized compound. Then from there, you do what is called target discovery. You look at a biological process that can be targeted for the treatment of a certain ailment. You do target validation. You use your pure compound, your compound that you have discovered. You try to see if it has effect on that target, call it target validation. Then you move to lead identification and lead optimization, where you try to knock off some functional groups of elements from the structure and see if it has better efficacy or better safety profile. Then from there you have a candidate drug or a candidate molecule. That is what drug discovery is about. And drug discovery over time has been immensely based on nature. About 50% of our current pharmacopoeia is based on derivatives of natural products. And these natural products can be from plants, microorganisms, marine organisms, endophytes, amphibians, reptiles, However, the competition in the pharmaceutical industry is very, very fierce and intense. And this has led to a decline in interest in drug discovery from the biodiversity in the big pharmaceutical industry, because natural product drug discovery became very expensive comparatively, time-consuming, and non-efficient. And a lot of legal issues about the bioprospecting started bugging it. And so most of the big pharmaceutical companies moved away from researches in drug discovery from natural products. And the fact that the natural products are actually big compounds, big molecules, unlike the small synthetic molecules. So most of the time it does not obey the Lipinski rule of 5. So a lot of time it tends to make the big pharmaceuticals look away from natural product as a source of drugs. Talking about the current status and what we are doing, in my laboratory, we try to look at drug discovery from biodiversity first by discussing with the traditional practitioners of traditional medicine. There's a WHO report that about 65% of the world population still depends on plants as their primary source of healthcare. And this, when you come to Africa, these statistics becomes about 80%. So it is a very, very important source of healthcare for us here in Africa. But there's also a need for us to begin to look at the pharmacological basis for the claims that are made for a lot of these medicinal plants or biodiversity, natural products from biodiversity. So we started looking at screening of the plants, the biodiversity that are used in traditional medicine for treatment of certain ailments. Did comparative studies to look at which is a solvent of extraction that will be best. And I'm particularly interested in typhoid fever. I've been doing a lot of work on typhoid fever. Apart from malaria, which is the most serious infectious disease problem we have in Nigeria, typhoid fever ranks second. And it's a very, very critical need for us to develop new and novel molecules to be able to deal with typhoid fever issues in Nigeria. So I've been looking a lot into typhoid fever, drug discovery for typhoid fever. And the starting point is usually from traditional medicine practitioners. Then we try to take these things to the lab to look for pharmacological basis for them. And we also know that the basis for most of the disease pattern in some infectious disease is inflammatory response. So we try to also look at anti-inflammatory activity of some of these things to see if it can help. And we've done a lot of studies in this line, chemistry of the plants, looking at its anti-typhoid properties and so on. We also try to do fractionation and to look for pure compound that may be the active principle responsible for the pharmacological actions we are seeing. And one of the things we also try to do now, which is a response to the problem that led the big pharmaceutical companies from working on drug discovery from natural products, is to have a very, very large library of compounds from natural products. And this is the flowchart for how we try to do it. I worked a lot with Ficus capensis and Eucalyptus camaldulensis. I'm just using it as an example here. We try to do pre-purification, use a preparation PLC to get several fractions, and look at the peaks on analytical HPLC. And once we discover we have pure compounds, we do structural elucidation, then we have characterized pure compound, and then library or a library of natural products referred to at a later time. And one of the things that we do from this is that we can then do bioinformatics studies or chemoinformatics studies on the natural product we discover. For instance, this fatty acid that we got from one of the plants, we tried to do some docking studies, and we discovered that it has binding affinity with a toll receptor 4. That is one of the main target for typhoid fever treatment. We've seen that one of the pure compounds we got from one of the plants has binding affinity for this toll receptor. So this can be a starting point for drug discovery. And so that is one of the ways we try to look at drug discovery from natural products in our laboratory as of now. Then we don't also want to stop at that. There is a need, we have discovered over the time that you can have some drugs or some candidate molecules that are very, very effective against certain microorganisms from a region. When you use that same molecule on the same species of microorganism that you got from another region, you see that there is a difference in activity. And we now want to look at the the molecular epidemiology of some of the infectious diseases in our country. And we are starting with, like I said, I have a lot of interest in typhoid fever, and Salmonella typhi is the causative organism. We are looking at trying to create the ecological niche map of Salmonella typhi in Nigeria. And when we do so, then we'll be able to address each region by region based on what they need is, so that we don't have cases of drug resistance. The substance we are trying to apply in one region may not be effective against the variant of the organism that is there. So it's one of the strategies we are also using in the lab currently. Then we can not talk about drug discovery now without mentioning COVID-19. The coronavirus pandemic just issue, and it's affecting us in Nigeria. The number keeps growing daily in Nigeria. And as a laboratory, we also started with looking for how we can find respite for our people suffering from this ailment. We have seen that the novel coronavirus is a self-limiting virus, meaning the major problem with the virus is the diseases that are associated with the virus, because the virus is self-limiting itself. Over a period of time, it goes. But the diseases or the symptoms associated with this virus are the major problem. And we see some of these symptoms like fever, fatigue, dry cough, a sore throat, shortness of breath, diarrhea, and so on. So what we try to do in the lab is to a way of managing these symptoms of COVID-19. If we can manage the symptoms of COVID-19, we believe strongly that we'll be able to manage COVID-19 itself, because the virus is self-limiting. And one of the things we are looking at now is to look at our biodiversity and look for products from our biodiversity that have antitussive properties, antioxidant properties, anti-inflammatory properties, analgesic properties, anticoagulant properties. Although coronavirus is said to be a respiratory disease, some researchers are showing that it's also a vasculotropic disease, and in fact it's a multi-systemic disease. So we want to look at all these things together: the immunomodulatory competencies, antipyretic properties, and biodiversity that's rich in vitamins. We look at all this, do extraction, do acute and subchronic toxicity studies. Of course, most of these things we are using are things that are already used as food or as traditional medicine. That's where we are getting it from. Things that our people already used to. So get those ones that have rich in all these properties and try to develop the COVID-19 therapy from them. That is one of the things we are doing in our lab now. Going to challenges. The challenges for drug discovery from biodiversity is both global and local. Global in the sense that the same challenges we are finding ourselves with in Nigeria is the same you find in India, in Indonesia, and in so many other places. One of the challenges in a lot of our biodiversity because of encroachments by people. Civilization is growing, the population is increasing, and so people need new places to settle in. And so cutting down biodiversity, we are losing a lot of some of our plants or biodiversity that are natural resources for drug discovery. So that is a major global challenge, and you can find it all over the world. Another problem that led to big pharma companies is in biodiversity is the fact that a lot of time there is extremely low yield. You take a plant, you discover that the amount of pure compounds you get is very small, and it can be very disturbing or discouraging for the farmer. Another problem is with limited supply. If it is a terrestrial plant, for instance, how many of those terrestrial plants are available? That is an issue. Then another problem is sustainable supply. Please just give me a moment. Let me take a sip of water. Throat is getting dry. Hello, sorry about that, please.
No problem.
Can everybody hear me? Another major problem
yes, please.
is yes, okay. With sustainable supply, even though the supply is sustainable, if we have to bring down some trees, for instance, to be able to get natural products from them, how long will it take for us to grow that tree back to get that tree back in nature? So these are problems. Then there is an issue of chemical composition. We have discovered that the same species of plants or the same species of biodiversity source may have different chemical composition depending on the region or the geography or the location of where you are getting it from. So you may have a plant that you are sure has this thing in your locality. You get that same plant from another place, you may not find that same chemical composition in the same plant. Another thing could be even the age of the plant. The age of the plant and the part of the plant you are getting, you are extracting from can affect the chemical composition. Another problem that is global is the complexity of chemicals from natural products. There are chemical substances that cannot be easily synthesized, and because it cannot be easily synthesized in the lab, it makes it very, very difficult to work with it using a high throughput screening. And there are the legal regulations of protect biodiversity in various communities. And because of this, it tends to frighten the big pharmaceutical companies from working on natural products from biodiversity in certain regions. These are global challenges. We also have local challenges for us that are very, very, very local to us here in Nigeria. And one of the major challenges we have is funding. Funding is a very, very big issue for us to do effective research from natural products that will lead to drug discovery. Need for funding sources in Nigeria. Another major problem is we find that most of our labs, so to do very, very research biodiversity, drug discovery from biodiversity, you have to collaborate with other labs from other places for you to be able to complete your work because you don't have the necessary equipment. Another problem is a problem of infrastructure, as we noticed. Even internet connectivity can be a problem sometimes, as we have noticed from this meeting today. Then light is a major issue. We see with powers for be able to carry out some research, some have to check some challenges we have with drug discovery from biodiversity in Nigeria, and also there's a need to train manpower in this regard in Nigeria. So these are the challenges we have in drug discovery from biodiversity, both global and local. Now, looking at the prospects, we have seen we have a very, very rich and untapped biodiversity. Very, very rich and untapped biodiversity. And this gives us an advantage in the sense that a lot of the drugs have in the market to actually build template of older that failed because, for instance, drug resistance. And when these newer templates come, it doesn't take time before drug resistance is established for these newer drugs. So there's a need for us to now look for novel molecules, novel compounds now that have not been in circulation before. And where is the best to get from? So there's a very, very rich and likely untapped biodiversity around Nigeria in particular that we can look to get this vehicle that we can use in treatment of diseases. So that one of that can be very, very helpful as a prospect for drug discovery from natural products. Then another thing is that if you one of the diagram I showed earlier, where I talked about drug discovery, the current status in my lab, we talked about prefractionation. If we can prefractionate our biodiversity, that is natural products from our biodiversity, and we have a lot of pure compound, then the problem of high trouble screening will be solved because we now have a lot of compounds to work with in the pharmaceutical industries. And this compound can also form a template for combinatorial chemistry to generate large numbers of analogs. You can use combinatorial chemistry or combinatorial biosynthesis to generate large analog of these pure compounds. So we can see that the nearest future, we can have very, very large source of new compounds from nature that can be used in the treatment of diseases. Then also, another thing we can look at, which is the prospect, is that the exclusive focus of antibacterial potential of medicinal plants and less attention to the ability of plant-derived natural products to boost the competence of host immune system to defend against pathogen attack in previous drug discovery research may not have been the most strategy. So we can change strategy now and start looking at how plants or natural products from biodiversity can be used for immunomodulation, which can now help boost the immune system to resist certain diseases. If we begin to look at drug discovery, can see that the natural product from our biodiversity can be a very, very rich source. In a modern drug discovery, a lot of emphasis is put on target identification and target several targets. And the fact that we can in silico method or chemoinformatic methods to do actual screening for our natural products, do indexing and such, can also lead us to have very, very good number of novel compounds for use in the treatment of disease in the nearest future. Also, if we engage in ethical bioprospecting, where the host community benefits from the drug that we discovered, as a pharmaceutical company will be benefiting, means that the host community will become more open into releasing their traditional knowledge for exploitation for drug discovery. So with the issue of ethical bioprospecting, you can have more collaboration between host communities and big pharmaceutical industries where drug discovery from these host communities can be established. Then there's improved technology. Technologies are improving over the years, and this can help us to also discover compound from biodiversity. There's also the change in perception. Initially, we're looking at some of the pharmaceuticals were looking at natural products from biodiversity that they may not be effective in the treatment of non-communicable diseases. In to fight or fend off other invading organisms. So because the non-communicable diseases are non-infectious diseases, that they might not be useful, but there are a lot of, in fact, a lot of drugs for cancer have been discovered naturally. This change of perception to improve research into drug discovery, natural. Then we can also begin to look at unconventional natural sources as sources. For instance, if you talk of the human milk, for instance, there are so many molecules in human milk that does not have any nutritional function. But we have not looked at them to see if they have. In fact, there is no known function for those compounds. But I don't believe those compounds can just be there without. So these are things we can begin to look out for and see if they could have pharmacological functions that previously explored. So we put all these things, we have international collaboration from one region to another. Then we see that the future is actually bright for drug discovery. Now, thank you very much. Thanks for listening. If there are any things, then I'll address the following questions. And thank you very much.
That's interesting talk. Lot of emphasis on biodiversity. The only thing is that we should try to protect biodiversity as well to gain something from them. And that is the problem we are facing at this time. Biodiversity conservation is required to be at the flagship conservation. It's interesting. Thank you for sharing us with your information. Is
Thank
Dr.
you very
Fayed there?
much.
Coming up.
Yeah, okay. So,
Sorry, that was very good. I just had to run.
no, that's okay.
I'm gonna give her. Please join me in congratulating.
Thank you, Dr. Musa. It was really nice.
Good. Thank you, sir. So I guess it's
Thank
open
you.
to questions, right?
Thank
Whoever
you.
wants to ask. I have questions, but I'll wait for you. Does anybody? I mean, this is a really great webinar with lots of. Geez, I don't know how to get out of it. Okay, I'll leave it alone. Please feel free. I know that there are several of you who want to ask questions. Please go ahead, and then I'll join in.
No question. That's an interesting talk. And the only thing is that the biodiversity conservation requires to be at the forefront of our movement. To save the planet, we have to save the product of the planet, and biodiversity is the product of the planet. It is a sum total of all species. So if we protect microbes to the mega species, then only our planet will be productive, remain productive. Otherwise, the problem we are seeing it, we lose the biodiversity of Amazon and other areas, and we get more problems. Somebody was saying that the COVID-19 is also because of that we are disturbing the forest areas and some viruses come from there, but I'm not sure. However, we need to share more information, and this is the diversity of the people we are listening from different continents. This time, somewhere it is night, somewhere it is day. So it is Dr. Nella Seth who have made us this possible, and I wish it continues.
Everybody pulled in this. I started it, but I couldn't go anywhere without everybody being part of it, and hopefully it will become much more. I know Professor Vijay Gunawardana had much to say in her webinar on this Ayurvedic dimensions. Nirmali, you want to say anything?
I like to comment, like Professor Dickson, you are very rich of your country with a lot of biodiversity, and thank you very much for enlightening us, to this part of world, how rich your country is with full of, resources. And I really appreciate, opening up our eyes on that particular region of in the world. Thank you, and lot of hard stuff you were presenting. I'm sorry, my background is not helping me to understand all these chemical jargons and chemistry and all. Thank you very much for your hard work, and I managed to understand so much things. Thank you very much, Professor Dickson.
Thank you very much.
Memo, I know you're interested in natural products. Do you want to say a few words, or do you have to leave? We can catch up later. This is recorded.
सूबा पंजाब, अगर आप ये समझ रही हैं कि चर्चिल की तरह या अब्राहम लिंकन की तरह इतना बड़ा लीडर आएगा, वो बैठेंगे और जिस तरह चर्चिल ने वर्ल्ड वॉर सेकंड के दौरान अपोजिशन से कुछ लोगों को मिनिस्टर बना दिया था, इमरजेंसी के हालात थे, वो तो हमारे मुल्क में नहीं है। आइडियली बिल्कुल सही बात है, ऐसा होना चाहिए था, कंसेंसस की कोशिश की जाती है कंसेंसस करने की, लेकिन जो ही ये मीटिंग से बाहर निकलते हैं, कराची से हमें रोजाना की बुनियाद पे आधी दर्जन के करीब जो प्रेस कॉन्फ्रेंसेस सुनने को मिलती हैं, आप जरूर गिनती होंगी। जवाब जो है वफाकी हुकूमत, उनके भी यहाँ पे वुज़रा यंगस्टर्स बैठे हुए हैं।
I think it's TV or the radio is on.
जो चीज़
yes. Thank you, Firas, and thanks, Dr. Musa, for such an enlightening talk.
For example,
I think it's a little late for me, so I might have to make a move, please. Thank you very much, all. Much
Okay.
appreciated.
Great. So Dickson, whose radio is that?
Thanks,
That's Hindi.
Firas.
I don't
Thanks,
know
Dr.
whose
Musa.
TV is that. That's Hindi.
Yes, that's Hindi. Yeah.
Yeah, absolutely.
Okay, Professor Yaya, you turn
Yes.
it on. It's
Yeah, I hear you. I hear you.
no, but it's coming to the talk. Everybody is getting. They can't communicate with it.
This is TV sound coming from somewhere. There is no TV in my house.
Okay, then it's Memo. Memo,
Yes. Yes.
Memo.
वैक्सीन की तैयारी के साथ साथ
You
लोगों
know
को
what?
बताया जा रहा है, लेकिन वो मुल्क जिसमें
Punjabi or Hindi or some Urdu or something.
ये चाह रहे हैं कि हमारे
Yeah.
Diversity of language.
Yes, it's
We have
we all-
500 of it in
Correct.
Nigeria, so it's not a problem.
So yeah, I'm trying to get him. Yeah.
कितना तरीके से हमारे यहाँ कमियां कुताहियां हैं.
Okay.
Hello.
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 Thanks, everyone.
So okay. Dickson, can you hear me? Okay, so I thought your talk was fantastic.
Yes,
I
I
have-
can hear you. Yeah.
Yeah, okay, see you guys. Whoever's leaving is fine. Just put a message and leave, right? You don't have to.
Yes. I too will leave, Professor Perez. It's late for me.
Okay.
Thank you very much.
Thank you.
Yeah.
Thank you very much. So Dixon, there are 2 or 3 questions. One is, I'm not writing a formal paper, but squash is a game we all share, and it's in a room, and it's semi-contact, and so it's considered one of the highest factors for coronavirus, COVID. So the Squash magazine asked me to write an article, and it took all my time. It's about 400 pages long. It's basically not an original article. I've surfed the web and put it together, and only the links and maybe some extracts from newspapers, some scientific journals, etc., etc. So I have a slightly better idea than when I was watching the news, and in that respect. So 2 or 3 things about that, and about plants that I thought might interest you. I don't know if I should present it or not, but I'll certainly share it with everybody once the editor, who's a co-author, approves it. But basically, that coronavirus attacks every tissue from head. First, let me back up a little. So it's only a small number of patients, like something like 20%, that are either symptomatic or lethally affected. 80% are asymptomatic. But in that 80%, there may be a very high number that is infectious. Highly infectious. In fact, the global numbers of infection and death are something like 5.6, kept by the Johns Hopkins University tracker, something like 5.6 million infections and something like 300 or 460,000 deaths. So, it's kind of basically, and when it attacks successfully, it goes through from head to toe. The really nasty. It's not just the respiratory system, the digestive system, its immune system, many places. And the virus mutates. So hopefully it will mutate into like what the flu pandemic did, and became, H1N1, which was an epidemic. It was not so lethal. Most of these viruses mutate. The vaccines, the fastest vaccine ever made took 4 years, and that was serendipitous, right? So I don't know how. There's a much bigger assault going on right now. And last of all, traditional methods of containment, right? In other words, isolation, masks, etc., the simple methods, had an fact
Yes.
of flu infections in the northern hemisphere, they were down. The flu season was shortened by 6 months, and the number of deaths
Okay.
was down by 60%. So those are
Okay.
measures put in place for coronavirus. They were having an effect on flu. So as far as
Okay.
I'm concerned, hopefully I will never have to be responsible for such thing. But if you are responsible, you go with the simplest thing that works well. And unfortunately, the ones who get hit hardest are the poorest and the developing nations because they can't control where they
That's
live,
true.
where the density of populations, they have no means, no money. So, this is going to be a global thing. They're actually talking about coming up with grants, large scale funding for that. So that's coronavirus. As far as the plants go, uh, I'm not a plant geneticist, I'm not a plant biologist, but I know from my early days that the soil, right, in graduate school,
Yeah.
the soil and plants, like this is work done by Virginia Walcott and others, Barbara McClintock, of course, was the one who was the grand founder of all forms of genome instability in all forms of life in her work done 30 years before DNA was even recognized as being heritable. She founded many, many things, and among them was that the genome was highly plastic. That means new kinds of genetic arrangements can come up. There's a new thing that you brought up and it's known recently that plants have a system of detecting which insects, uh, might be hostile and bring
Yeah.
chemicals to hit them, and which insects they want to attract for pollination, right? So, and then there are many other immune factors that they found, and they can't walk away like animals can, so they have to have a good immune system.
Yes.
So since Virginia Walcott's time, it is known that the genome is unstable, and that the anions and cations in the soil will affect the stability and the amplification of specific sequences like ribosomal DNA. So let's say
Yep.
you have variation of these plants isolated from different geographical locations. If you grew them in a
Yep.
chemically homogeneous position, you might risk losing some of the diversity and some of the properties that comes from natural systems. But you can make them more uniform, perhaps in their immune responses or in their chemical repertoire, and so select for each plant for reference. Just a suggestion. There are many other
That's
things. The talk was loaded with stuff. I'm amazed at how much you got done and how many places. I really want to congratulate you. It's really a privilege to have you in our institute and managing to stay so active in the institute's business despite having this active lab. Thanks very much.
Thank you
Anybody?
very much.
Yeah. Uh, Anybody else want to say things, or you have?
Maybe I can respond to an earlier comment that talked about conservation of biodiversity. Like I said in the talk, anthropic pressure is a problem for those of us working in drug discovery from biodiversity. So we are also in the forefront of conservation of our biodiversity because those biodiversity has to be there for us to be able to get the natural products from there. So conservation of biodiversity is very, very integral to the type of work we do. Thank you.
Okay, so I don't know who is next on. Let me just check very quickly. So we know, we have at least a month, and institute. So one more thing, why don't we, while we are here, everybody? So I'm just going to look very quickly at put this up on the webinar schedule.